The overall goal of this protocol is to demonstrate how to combine the use of microfluidics with microelectrode arrays to generate a platform suitable for studying neuronal communication and axonal signal propagation.
Hello, I'm Paulo Aguiar, and I'm the PI of the Neuroengineering and Computational Neuroscience Lab at INEB, the Institute for Biomedical Engineering at University of Porto, Portugal. Understanding information transmission in neural circuits remains one of the biggest challenges in neuroscience. Today we are going to show you how we can combine in vitro microelectrode arrays with microfluidics and software tools developed here at our lab to detect and characterize propagating action potentials. We use this setup in a variety of studies, namely neural coding and decoding, and in the communication between sensory neurons and other cell types.
We believe that the visual demonstration of this protocol is crucial, since microfluidic and microelectrode array assembly is technically demanding and difficult to reproduce just by reading protocols. Furthermore, culturing cells on this platform is not straightforward as in conventional MEAs, and it requires special attention to the cell seeding step and culture maintenance.
This setup allows us to study several communication-related properties, such as propagation velocity and direction, in a well-controlled environment. Although the recording procedure is simple, data analysis requires knowledge and the use of the right computational tools.
On the day before cell seeding, start by preparing the microfluidic devices by cleaning them with vinyl tape. Gently press the tape against the device to reach all areas of the microfluidic.